Okay, hey guys, so this is going to be the explanation for the PowerPoint of Lesson 3 for Genetics, which is 375. Okay, so here are the lovely goals for the section. Examine the difference between all the different sexual inheritance patterns and understanding the importance of reciprocal crosses, examining the features of X-linked and Y-linked and pseudo-autosomal, introducing epigenetic inheritance, examine how X chromosomes inactivation and genomic imprinting can affect phenotype, describe the mechanisms of X chromosome inactivation and genomic imprinting. So moving right along here. So sex limited is only observed in one sex. So it can only be females or it can only be males. So right here it says that males and females can have identical genotypes, yet the male or female displays the phenotype only. So that kind of explains what sex limited is. So moving right along. So here is sex influenced, which we talked about if you watched the practice problems for lesson 3. So here we're shown baldness in males and females. And so for females, the capital B is actually going to be recessive. So they need 2 capital B's in order to be bald. And so if they only have 1 or if they have 2 little b's, they're going to be non-bald. But the big B is actually dominant in males. So if they have heterozygous or dominant homozygosity, then they're going to be bald. So that's what sex influenced is, when it's recessive in one and dominant in the other. So sex-linked inheritance is going to be the X and Y chromosomes are distinct in appearance and carry different genes. So we can determine if an individual inherits a particular sex chromosome by their sex. So we know if it's XX, then it's a female. If it's XY, then it's going to be a male. Okay, so now we're going to start talking about Drosophila. So the wild type is going to be this red eye here, and the mutant is going to be this white eye. So we know that Drosophila is X-linked because when a cross was done between a red-eyed female and a white-eyed male, they got only red-eyed F1. And when they crossed the F1 together, the F2 generation came out with 1 white-eyed and 3 red-eyed. And so, right here, they crossed a red-eyed and a white-eyed, and they got half and half. So, X-linked inheritance, because no white-eyed females were observed in the F2 generation, and inheritance of the eye color trait depends on the sex chromosomes present, we know that the eye color trait is X-linked. So these are pretty nice little test crosses here. It's good to look at those if you want to pause the video and check those out. So now we're going to talk about X-linked inheritance reciprocal crosses. So this is when we take reciprocal crosses, meaning that we cross a homozygous dominant female with a recessive male. And then when we cross those, X-linked, if it's X-linked, it'll give us reciprocal crosses with different phenotypic results. But if it's autosomal, the crosses will give the same phenotypic result. So you can see that here. Okay, now we're going to talk about hemizygosity and Y-linked alleles. So males are hemizygous for the X and Y chromosomes, meaning that they have both, and X and Y linked genes. Holoandric genes are those that are present on the Y chromosome only, so they can only be passed from father to son because women don't have the Y chromosome. Okay, so pseudo-autosomal inheritance is going to be this MIC2 gene here is an example of pseudoautosomal. So the alleles are on the sex chromosomes, and the inheritance pattern is identical to the genes found on the autosomes. So you can see that here and here. Okay, and epigenetics is a change to a gene or chromosome that influences the expression of the gene. So right here, so here we see this cute little calico cat. So here are two types of epigenetic inheritance. Dosage compensation occurs in embryogenesis and allows females and males to express similar amounts of X-linked genes. So genomic imprinting occurs during gametogenesis and influences the expression of paternal or maternal genes or maternal alleles of a gene in the offspring. So this coat right here is dosage compensation that allows females and males to express similar amounts. Okay, so looking further into dosage compensation here, is the level of X-linked gene activity is the same in males and females, so we know that there are equal numbers. One of the X chromosomes in females is inactivated, and in fruit flies, the expression of X-linked genes in males increases twofold. Okay, so finding evidence for X inactivation. So two lines of evidence suggest X inactivation. So the studies of Murray Barr and Ewart Bertram, and they found Barr bodies, which we'll talk about a little bit later. So the studies of Mary Lyon found that female cats heterozygous for X-linked coat color show a mottled coat, or this calico pattern here. Okay, so looking further into the Lyon hypotheses, so she was looking at black coat color and white coat color in mice. So her hypothesis said that during embryogenesis, both X chromosomes are active. And also in an individual embryonic cells, one X chromosome is randomly inactivated. Okay, so females heterozygous for X-linked alleles are mosaics, which means that they'll display this neat little pattern here with patches of different colored fur. Okay, so this X inactivation is pretty neat, actually, I think. X inactivation center, I call it the XIC, I don't know if that's correct or not. So mammalian cells can count the number of X chromosomes. So in females, they can see that there are two X's and one of them is inactivated. So there are certain diseases where people have too many or not enough sex chromosomes. So here in Turner syndrome, you can see that there's only one X here. So in XO females, where there's only one X, the X remains active, so one of them is not inactivated. So in females with three X's, two X's are going to be inactivated because we only need one to be active. And in XXY males, one X is inactivated because they need one X and one Y. Okay, and so this XIC right here actually counts the numbers of the X chromosomes. So if there is more than one XIC, additional XICs and X chromosomes are inactivated. Okay, so now we're adding in this XIST and T6 and XCE, which is what is involved with this XIC. So the XIST is active on both X chromosomes, and it coats and inactivates one of the X's. So it's pretty important. You need it in order to inactivate the X chromosomes. So T6 targets XIST for destruction. So that is what causes Barr bodies. So here, X chromosomal controlling element, XCE, is a binding site for the proteins that regulate transcription of both this X's and T sex. Okay, so this is a pretty neat picture. If you want to pause this, it's just kind of what we just talked about here. So let's see. Genomic imprinting. So this is going to be talking about an IGF-2 gene. So genomic imprinting is when genes are inherited from one parent that can be chemically tagged by genomic imprinting. So this tagging causes the offspring to only express one of the two alleles, which is called monoallelic expression. So this gene we're looking at is body size in mice, which is either dwarf with the 2 negative or normal size with just the regular 2. Okay, so this is a pretty neat little thing here. So establishment of the imprint. So this is showing that this is going to give us the imprint right here, as well as this right here is going to give us the imprint. So then when it gets put into the mouse, here and here, we can see that all of the gametes are going to be 2 minus here. And then all of the sperm are going to be just the regular 2. So they're going to be the dominant. Thanks for watching.